Well, good afternoon or good evening, everyone. So I'm very pleased to see you here. And now I'm going to bring you to a biological world. So today I try to tell you something which we have been working on and integrating microfluidics and optical technologies for single cell study. So I'd like to give you some background. So why we're interested in study single cells? Well, this is to study biological population at the single cell resolution. So we all start off as a single cell, and we all different. And even for this individual, for the identical twins, they all have different features, such as the fingerprints. And we all know now this individual heterogeneity exists universally, and it can be essential for cell survival. I give you one example, such as a superbug, and these are a few number of cells that survived intensive antibiotic treatment. So everyone know now antibiotic resistance become a serious health challenge globally. And now in traditional biological labs, we culture cells in flask and we measure cells in this 96 well plate. There are millions of cells in each well. The results we get are endpoint results, and these are average response of a whole population. This measurement doesn't necessarily tell us the true story. For example, if we're interested in GFP reduction, right, this is a protein, it can come from the top scenario, yeah, or it can come from the bottom scenario where the half of population lost the ability to produce the GFP. So this population approach and give us a rough idea, but we are not able to study it in details. And traditionally, isolate cells is necessary for most of biological studies. And this is done through serious dilution cultivation to form these individual colonies and in order to generate a population. This is time consuming. And in clinical diagnosis of pathogens and to determine whether the antibiotics is useful or effective, they always will conduct this antibiotic susceptibility test, which we short for AST. Normally, it will take at least 2 days. During this waiting time, yeah, seriously ill patient can die. So the way to speed up this diagnosis is to remove the need for culture a large number of cells. And that is what we think single cell approach can be a promising method. So this is where microfluidics come to play. I just give you a very brief introduction. So microfluidics device consists of tens of microchannels. At this length scale, it matches well with the biological cells. In this way, we can precisely manipulate individual cells. The results here, so this example here show you that we can use microfluidic device to generate larger scale cell array, and we can monitor how cells respond to drugs. And the right example shows you we can even trap individual cells in this droplet, allow us to perform high throughput screening. But when we come to talk about the bacteria, they're very small. There's around like 1 micron, and they move around. So it's quite a challenge to use microfluidic to study single cell bacteria. Now, in the past few years, there are quite a lot of examples, good examples, such as mother machine, and these have been developed to study single bacteria cells. But overall, the microfluidic device for single cell bacteria study is still underdeveloped. So in our lab, our approach is interdisciplinary approach. We use a microfluidic device to manipulate cells as well as their microenvironments. we integrate microfluidic device with advanced cell measurements, and this part we focus on label-free direct measurement of living cells. And this, today I'm going to focus on imaging-based approach as well as Raman spectroscopy-based approach. So let us give you some example, as I just mentioned to you, that traditionally antibiotic susceptibility tests take a long time because it requires a large population of cells. So a while ago, we started to look at this problem and try to explore a single cell approach to develop a rapid AST test. Now, AST tests need to test a wide range of concentration. We can't just use this simple gradient microfluidics. On one chip, we are able to measure a wide range of the concentration. As you can see here, this is stable gradient concentration, and cells are trapped as a molar layer on chip, and the location of a cell corresponding to a specific concentration. And because we trap cells as a molar layer, now through this time-lapse imaging, we are able to monitor the clear cell images. We can correlate the change of cell structure, cell area with cell mass, and thus allow us to determine the cell growth rate as well as the lag time. Okay. Okay, so as well as this lag time. And I would like to mention the lag time normally is not able to determine using the traditional method. So, of course, for any new approach, we need to validate with the traditional method. As you can see here, for the minimal inhibition concentration determined from the golden standard, and the single cell approach matches very well. Now, the advantage is with a single cell approach, we are able to complete AST test within 5 hours. Not only that, now we are looking at the whole population at the single cell level, and that allows us to observe all kinds of behavior. So, and this is one new phenomenon which we observe. Some cells, right? If for example, the cells here, if they have ability to utilize the dead cell from nearby, they gain the resistance to the drug, and we call this one opportunist persistence. And that will tell, this single cell approach really resolve a lot of new information. And very soon, we developed a series of single cell microfluidics. And this is one example. Now, the example here I show you, so we have many chambers. We use the middle chamber to trap cells into molar layer, and we use the side channel to deliver different reagents, different cell. That will allow us to control the microenvironment in a flexible way. Similarly, we use the method I just described to correlate cell area with the change of the mass, and we can obtain the cell's growth rate as well as the lag time. And now we have a lot of data from individual cell. So what we can make use of this rich individual cell data now, we have now got a new means to look at heterogeneity of population. Look at this 2D plot. Individual dot represent a cell, and we obtain the lag time, we obtain the growth rate. So the area of this cloud will tell us whether this population is more heterogeneous or is more homogeneous. And that can allow us to determine how the whole population responds to stress. Similarly, because there's so many individual data, we can easily get an average value for the population. We can also determine the distribution of the population change. Yeah, so here we can look at whether the stress to make the more sick cell die or to make everyone subdued. So that gives us this information, the hidden information. This approach can be used for many fields, and it is in particular valuable to study antibiotic resistance and how antibiotic resistant gene transfer. I just give you one example. For example, here, now we know that the antibiotic resistant gene spread, and that is the current really a serious concern. We use a donor strain. Yeah, this donor strain it expresses red fluorescence, so we can see it, and it also contains the resistant gene to antibiotics. If the donor cell can transfer this plasmid into recipient cell, okay, so here the horizontal gene transfer occur. So this is normally the first instant for antibiotic resistant gene to transfer. The recipient cells is going to express the green fluorescence, okay. Now, you also know, as I just mentioned, cells were produced that will divide, which means that this transconjugate will divide. If we are going to use the traditional method, these endpoint measurements, we are not able to correlate the number of these green fluorescence cells with the amount of this HGT frequency, okay? What we're able to use the single cell microfluidic is to monitor when, where all this HGT happen, and when and where that this vertical gene transfer, yeah, due to the cell growth happen. So we can decouple the two fundamental process apart. And what we can use that one for, that is very interesting. Again, a lot of discovery. In particular, we look at how antibiotics treatment affect this antibiotic resistant gene spread. And we find that actually the donor, how the donor resists to antibiotics play important role. Look at this figure here. This donor cell can resist to the TMP. Okay, TMP is one antibiotics, but susceptible to other antibiotics. Now, as you can see from this figure here, if we treat the whole population, yeah, and in the presence of the TMP, you will find the antibiotic resistant gene spread much faster and more than when we use another antibiotics that suppress this donor. What does that mean? It means if we have this antibiotic resistant bacteria in our body, if we are not able to correct the diagnosis, what kind of drug should be used, we will make our situation even worse. And also it's interesting because we have so many individual data, we actually find the spread of antibiotic resistant gene is like the spread of virus. So we use the same model to model how the antibiotic resistant gene spread. And of course, we use the system for many other applications, and these are the publications. And in the interest of time, I'm not going to elaborate. Okay, so that is one part of the story. Now, we should look at there are many microorganisms in the environment, in our gut, right? And the majority of them are not culturable. So we term this type of organisms, so we term them as a dark matter. Yeah. But similarly, very often, a rare cells in the dark matter that play important role, they have a key function. So what we will do. So this comes to my second part of talk, the Raman spectroscopy-based technique. First, I give you a very brief introduction of Raman. It measures the vibrational energy of chemical bond, and therefore it reflects the intrinsic information of chemicals. Because of that, the molecules, even they're quite similar, yeah, they have similar structure, they will show a very different Raman spectra. And that is why we call Raman fingerprints. The problem is that Raman, this inelastic scattering is quite weak. So the Raman signal are normally generally quite weak. Spontaneous Raman signal are weak. And for bacteria, because it's small, we can acquire a spectra from the whole cell. And the single cell Raman spectra contains of more than 1,000 bands. And that tells us the features of all the biomolecules, such as nucleic acid, protein, and lipid. Based on the single cell Raman spectra, we can classify what type of cell is. We can classify the physiological states of a cell. And if we're interested in whether the cells can use a certain compound, and that is metabolic activity, we can use this called stable isotope label to probing the cell's metabolic activity. And I'm going to give you one example. For instance, if we're interested in whether cells can use glucose, just in example, we can use a carbon-13 labeled glucose. Any cells that are able to utilize this compound, carbon-13 compounds, they generate this shift in their single-cell Raman spectrum. So that is a criteria for us to classify, identify these target cells. Okay. So the there, of course, there are many other applications using stable isotope labeling. Now, we have now got means to classify the cells, and the important thing is how we select the target cells. And we know that traditionally, fluorescence active cell sorting widely used. But this technology, we need to label cells with fluorescence, and we need to know what kind of what part of cells should be labeled. So this requires a very well characterized system. But as I just mentioned, many microorganisms are dark matter. We have a limited knowledge about it. So won't be good if we use the Raman spectra as a readout, but sort cells in a similar manner as FACS. I also mentioned that the Raman, spontaneous Raman is quite weak, which means, right, it's quite a challenge to detect it when cells are in flow. So originally, and the first Raman active cell sorting is done through the optical tracing, and then since then, there are several developments in this field. The common challenge we still face is the throughput, because you have to trap the cell and wait and to then detect it and collect it. Of course, it's a problem if we want to isolate rare cells. And we have to go through a large number of cells. So ultimately, if we can sort the cells in a similar manner as a FACS, that is the way to generate a high throughput sorting. In order to do that, we have to overcome the problem I just showed you in this video. Because the signal is weak, we use the low flow regime, and the downstream sorting will affect this detection area. So we have to generate a very stable detection, Raman acquisition. And this comes to the work we've done in our lab. So we design a quite a simple pressure divided chip, and here we use a high resistance to eliminate the fluid actuation around this detection area, and that allow us to deliver cells precisely to the detection region in the presence of the sorting, and that allow continuous Raman active cell sorting. So of course, in order to achieve high throughput, we need to achieve the automated operation. So here is the interface of the control software. You can see, so here we detect the target cell, always constantly acquire the Raman signal. The first system we demonstrate is using this the carbon-13 labeled, to look at whether these algae cells, can utilize carbon-13. So we based on this stable isotope labeling. That time already 96% purity, but the throughput is quite relatively low. And the advantage I wanted also to highlight, because it's under the flow, the system, which means we can precisely control how long each cell's exposed in the laser, which means we can control the overall laser intensity to each cell. And as you can see from this system, from this imaging here, that if the intensity is too high, cells will die. Yeah, so the flow system will have no detrimental effect on cell, which means the cell we collected are viable. Now, the advantage of the Raman spectroscopy is that we have a lot of information, and it allows us to do the unsupervised classification. Our Raman active cell sorting system also function as a flow cytometry, which allow us to obtain thousands of data. We can use this one as a training data to using the machine learning algorithm to generate a classify, and that will allow us to classify the cells without any pre-knowledge. So, but we have to consider that this is on-the-fly classification, right? The computation time is important. So here we use the LDA and to classify the spectra, and then use that one as a criteria to sort cell. Very soon we realize actually when we started to look at the dark matter, when we start to look at the environmental microorganisms, they are very complicated. That the community consists of all kinds of size so that it can block a chip. Because of that, we further develop this new 3D flow focusing RAX system. It consists of a large detection chamber around 1 millimeter, and we effectively use this schist flow to deliver all the samples into this focal point, which is only 3 micron. Yeah. And this is remarkable. Consider the chamber is 1 millimeter in diameter. Of course, we also integrate this optical fiber online that will improve both the throughput as well as the accuracy. Now, the additional advantage that is with this large detection chamber, we remove any interference signal from the device. We all know, right? If we can eliminate the noise, we actually improve signal to noise ratio. And as you clearly see here, because of that, we're able to acquire a weak Raman signal from samples such as E. coli. So we use chlorella and E. coli. So this is two different species. We use this one as an example, and we demonstrate this new 3D system, right, can clearly sort all the mixture with a different size from 1 micron to tens of micron, and it has a really reliable operation as well as a high throughput. At the moment, this is perhaps the high-end throughput for the for cell sorting based on spontaneous Raman signal. So in a nutshell, the Raman active cell sorting, this flow-based system can be universally applied to many different systems. It has an advantage in terms of high throughput and can be further integrated with enhanced Raman techniques such as simulated Raman spectroscopy to further enhance the throughput. I have mentioned to you, flow-based system has no effect on cell viability, so the collected cells can be used for further culture or do the genetic sequencing. And this is very important because it allows us to link phenotype function with genetic identity of individual cells. So here a simple conclusion. So I have shown you the single cell microfluidics allow us to monitor individual cell in real time, and so that allow us to discover hidden diversity. And we have shown that this approach can lead to many new discovery. The VAX system at the moment is still at the early stage. However, I hope that I convince you that the Raman active cell sorting has a huge potential to look at microorganisms, in particular for the environmental microorganisms. I'd like to thank the people who did the work, yeah, as well as my collaborators, in particular Professor Wei Huang at the University of Oxford, and here the founding body. And I'd like to thank you for your attention, and I'm happy to answer any questions.
Thank you very much. Do you have questions? Nanjing as well, yes. See Nanjing. Do you have questions from Nanjing?
Yes, I have a question. For your CS, what's your wavelength of your lasers?
And that is 532 nanometer. Yeah. But for mammalian cells, it's better to use a bit toward the infrared. For example, 785 nanometer will be better. It can suppress the fluorescence, in particular autofluorescence from the sample. Yeah. And has less damage to cells.
I was going to ask about the intensity. You told us what the energy was, but that needs to be delivered in a short pulse.
Exactly. Yeah. That is excellent question. Very, very good. Yeah. So if we can deliver a short pulse, and we will be able to get a high signal. At the moment, we have to use a very low intensity. Otherwise, we damage the cell. But if we are able to use high intensity short pulse, and we will get a high Raman signal, and also that will eliminate any problem to the cell viability. That is a much better way. And that is very expensive. If people have this nano, what do you call it, nanoseconds laser, that would be better.
Yeah, can I ask a question? Because you mentioned the pulse laser, right, actually. So there's another new technology called the CW stimulated Raman, right?
Yes.
So currently you use the spontaneous Raman because the signal is weak, and also because the cell is difficult to use the expensive nanosecond laser or picosecond laser. Exactly. Yeah. Is there any thinking about the user's continuous stimulated Raman technology? There's a continuous laser stimulated Raman.
All of what we are doing is a continuous Raman. So we're using the continuous laser. The stimulated SRS Raman spectroscopy is using, you have to using the pump.
I know, we use the two one
exactly.
That is the signal
that is very expensive.
Yeah, but that's new technology for the continuous laser one. There's no pulse laser one. So there's another technology, use a cheaper laser diode from the CW laser, continuous laser to do the stimulated Raman.
If that can be achieved, that would be fantastic. If that can be achieved, there are a lot of advantage in terms of high throughput, right? And that would be fantastic.
We have a good idea, right? Probably potentially we can.
Okay. Yeah.
Thank you. Very good.
No problem.
Take the photo all together. Yeah.